Okay, so now that we've talked about some of the structure of antibodies, let's focus in on some of the properties of antibodies. And really, we're just focused here on the high yield information. So looking back at our antibody structure here, we can see again, we have our variable region that's going to be up here in the pink color. And then we also have this constant region that's down here in the blue color, as we just talked about. And this is functionally how we want to kind of think about dividing the structure. And again, this variable region, it's going to contain the area that we'll call the idiotype. Okay, so the idiotype is going to refer specifically to the binding of this antibody receptor to some epitope, right? So that's some region on our antigen. So the idiotype being this structure here. And again, the constant region representing the area where we have our isotype. The isotype representing what type of antibody do we have? Is it going to be an IgM antibody, an IgG antibody, etc.? Okay, so we have the idiotype up here, the isotype down here, again, the Fc region, and then we have the FAB, the antigen binding region up here. Now remember that this can also be divided based on the types of chains that we have. So we have a heavy chain, right? So the heavy chain is going to be this chain right here down in the middle. And then we also have a light chain, okay? So the light chain is going to be this one that's on the outside, only in the antigen binding region, the FAB region, right? So that's how we separate this. And again, the thing that you want to remember is that there's different genes when we're talking about our gene transcripts that kind of get shuffled around randomly to create a whole bunch of different idiotypes so that we can bind to all types of antigens, okay? And those shuffling of genes is sometimes referring to specific segments here, VDJ recombination. And you can see that this D is in parentheses. The reason for that is because the heavy chain segments use the VD and J shuffling, whereas the light chain segments primarily are going to use the V and J shuffling. So this is just a little thing just to be familiar with. Again, it's these little details that sometimes come up. But the thing that I want you to kind of take away from this is that this whole process of VDJ recombination, if you think about it, right, you have some B cell, and inside of this B cell is going to have these genes with the V segment, with the D segment, with the J segment. It's going to have those genes inside of the nucleus, and it's going to shuffle those genes around until we get some unique immunoglobulin receptor, right? So we shuffle all of these genes around, and then we come out with this very unique idiotype on this B cell receptor. Remember, what is a B cell receptor? A B cell receptor is essentially a membrane-bound antibody, right? So it's like we created an antibody, like we see here, we just stuck it on the surface of a B cell. Specifically, IgG and IgM are going to be kind of notorious for being on these membrane-bound surfaces for B cells. You can also see this with IgD a little bit, as we'll talk about. But the point is this VDJ recombination is going to happen in these B cells, but there was no antigen that stimulated this process. This process happened as B cells matured. When B cells were maturing, where are they maturing, by the way? They're maturing in the bone marrow, right? We talked about that. While these B cells are maturing in the bone marrow, they're shuffling these genes around, these V, D, and J genes in the heavy chain and the V and J genes in the light chain, and they're making unique idiotypes, right, that are going to go on the variable region in the fragment antibody portion that we just talked about right up here. Okay, so that's what this whole process is. Again, the big thing I want you to remember is that this is going to be antigen independent. Okay, so there was no antigen that stimulated this process. It's just going to happen as B cells mature. We're going to make a whole bunch of different receptors, right? That's the point. And that's why this is completely random. It's random recombination of these genes. But you got to remember, we're doing this amongst millions and billions and etc. B cells, right? On a mass scale. So we're going to get all different kinds of receptors. And ideally, certain receptors are going to favor the binding of future infections, right? So that's the concept. And this is all going to happen in the fragment antigen binding portion up here, which is where we're going to bind those cells, right? The constant region, we're not binding antigens in the constant region, right? The constant region we talked about in the last video, it's about complement binding, it's about opsonization, phagocytosis, etc. So that's the concept here. Now, when we go into this next category here, somatic hypermutation, affinity maturation, isotype switching. So these are things that we've also kind of already hit on a little bit if you've been following the series so far. And really, somatic hypermutation, essentially think of this as, once we've interacted with that antigen, so we go back to our B cell here, once we've actually kind of found our antigen, so let's just say that we have some, we'll draw some pathogen here, maybe it's a bacteria, right? So we have some pathogen here. And again, on the surface of this pathogen, we might have an antigen. So let's just say that there's some antigen here, okay, some antigen here. And then our B cell receptor might interact with that antigen. It might be the perfect fit, right? We might find our epitope here on this antigen that this B cell binds to, and it's a perfect match. And then what happens, right? These B cells can potentially get activated. Remember that B cell can then kind of internalize some of that antigen potentially, and then redisplay that antigen in MHC2 to then activate T cells if this was a T helper dependent response. If it was T helper independent, this B cell might just turn into a plasma cell potentially and release IgM. Okay, so these are all things that we talked about in the B and the T cell video. But again, this process, once we activate this B cell, these B cells, some of these activated B cells can go to lymph nodes, right? And they can go into those secondary follicles we talked about initially, those pale staining germinal centers, and they can proliferate. And so you get a whole bunch of B cells proliferating in the lymph node, in those germinal centers. And as they're proliferating, what these B cells are doing is they're essentially saying, look, I know this receptor binds to this antigen, but what if I just tweak it a little bit, right? Maybe I'll just add maybe an extra couple peptides here or amino acids here. Then let's see if it binds more tightly or more weakly. And essentially the B cells are doing this in the lymph node on a very large scale. So they're randomly mutating the known binding receptor and trying to see if we can get it to bind even stronger. And those receptors that do bind the strongest survive. And that's called affinity maturation. We're essentially maturing the affinity of the receptor. We're finding the receptor that binds the tightest. And how do we do that? Well, we do random variations, again, in the variable region. Same region, right? We're just making little tweaks to the variable region that we already know binds to the antigen. Now, that's different from VDJ recombination. VDJ recombination, we're just mixing up stuff. We don't know what we're doing. We're just shuffling genes everywhere, and we're just trying to make a diverse pool of B cells. With affinity maturation, we're looking for something that is already bound to its antigen. We just want to make that binding stronger. And for that reason, this is going to be antigen dependent. Okay, so this is going to depend on the antigen that binds to it. That's what's going to trigger this whole process. So somatic hypermutation, affinity maturation, here, let's kind of highlight these. These are going to be antigen dependent. Okay, isotype switching is as well, which I'll talk about in a minute. And then VDJ recombination, that's antigen independent. Now, why is isotype switching antigen dependent? Well, you can kind of put the pieces together based on what we've already talked about. So remember, the whole point of isotype switching is after this B cell gets activated, what is it going to do? Well, like we just said, it's going to internalize this antigen, right? So it's going to take that antigen, it's going to internalize it potentially, and then after it internalizes this antigen, it can display that antigen on MHC2 receptors, because remember, B cells are antigen presenting cells. So let's say that there's some MHC2 receptor here, okay, so the B cell can then display it, and this is really the same song and dance that we keep talking about. So this B cell then is just going to go to a T cell. What's that MHC2 receptor going to bind to? It's going to bind to a T cell receptor, CD4 co-receptor, right? 4 times 2 is 8, rule of 8s. So that B cell, that's an antigen presenting cell, then presents this to a T cell, then it becomes activated. And that T cell will also have this CD40 ligand that binds to CD40 on the B cell. Remember, this is particularly high yield, and we've talked about this a couple times. So that binding between the B and T cell is going to stimulate class switching. Okay, so we have kind of two receptors binding, MHC2 binding to the T cell receptor and the CD4 co-receptor, and then we have CD40 ligand binding also, like we talked about before. And if this is unfamiliar, go back, check out those previous videos on B and T cells. We'll go into this in greater detail, but essentially that binding is going to stimulate class switching, and then we're going to have certain cytokines that are released that'll stimulate different versions of class switching. If we want to make IgE antibodies, right, we might see interleukin 4 and interleukin 13 being released. If we want to make IgA antibodies, we might see interleukin 5. So again, you want to remember these specific cytokines as they're particularly high yield to note in this isotype switching. But going back to my original point, how come this is antigen dependent? Well, it took an antigen to start with to get this whole process going, right? We had to internalize this antigen, display it on MHC2 to eventually present it to a T cell to get isotype switching. So that's why both of these are antigen dependent. Again, VDJ recombination, antigen independent. Now, the big difference between these two is that somatic hypermutation, affinity maturation, we already said it's taking place at the variable region, right? It's taking place at the variable region. We're slightly changing this idiotype so that it gets the strongest binding to our antigen. Whereas with isotype switching, what we're doing is we're changing the constant region. So if I change the constant region, what can I change? I can change it from IgM to IgG to IgA to IgE by using these cytokines, right? So that makes sense that we'd be changing the constant region in isotype switching. Now, the last thing I want to talk about here is the primary response versus the secondary response. And this is actually pretty intuitive to kind of understand. So let's just say that this is time, and then let's just say that this is antibody concentration in the serum. Okay, I'll just put an S there for serum. So this is my antibody concentration in the blood or the serum, and then I have time here on the x-axis. I'm going to move it over so we can see it better. Okay, so this is time, and then we have antibody concentration in the serum. Now, let's just say that at some point here, we are infected with some antigen. Okay, so we'll just put a 1 here to signify that this is the first time that we've seen this antigen. Maybe it's a bacterial infection. Okay, so let's just say that we have some bacterial infection. This is the first time we're seeing this antigen. So what will happen is initially we might have some stable level of IgM that spikes. Okay, so our IgM level, after realizing that we're infected with this antigen, again, we're going to have an inflammatory response, and some B cells will recognize this antigen for the first time and turn into plasma cells and increase the amount of IgM being released. Okay, so we'll get some IgM release here. So we might also get some IgG being released. And the IgG response we'll get after class switching is actually going to be much greater than that initial IgM response. Okay, it's going to take B cells a little bit of time to undergo affinity maturation, isotype switching, but eventually we're going to generate an even stronger response using the IgG, all from that first exposure. And now what will happen is, remember, after we have an initial exposure, there'll be memory B cells that are around for future events with this antigen. And let's say we get infected with that same antigen bacteria a second time. Okay, so the second time, what's going to happen to the IgM concentration? The IgM concentration is going to do pretty much the exactly same thing. Okay, it's not really going to change much, right? The peaks here are about the same. Okay, and the amount of time it takes for IgM to respond, it's pretty much all going to be same song and dance. IgM doesn't involve class switching. It's kind of like your standard antibody, right? Even the T helper independent responses will still release IgM, like we talked about in the B cell video. However, the difference here is that now we have some of those memory B cells that are around, and they've already undergone class switching, and they have very strong affinity maturation and high levels of IgG that can be produced. So what will happen is we'll get a slightly faster and much stronger response from IgG. So I'm just drawing a massive jump here in your IgG response. So we have an exponential increase in our IgG response this time. And that's because we have memory B cells that have already undergone this whole process, really, right, from some kind of T helper dependent response that are now generating, significantly more amounts of IgG. And specifically, that IgG is going to come mostly from plasma cells, right, from this memory response, okay? So again, the key is on that second exposure, you're not really seeing a huge change in IgM in terms of timing or the absolute concentration of it in the blood serum. However, on re-exposure, you can see there's a lot more IgG that can respond actually slightly faster even potentially. And this is where you see things like, if you want to see if this person was ever infected with a certain bacteria, you can try and draw their antibody to that bacteria. Or if you vaccinated somebody, you can see their titers, right? You can see how much IgG, how much of a response they generated when they were vaccinated. And so you can see if they're immunized by having a set level of IgG after re-exposure.